Hello, am I audible to everybody? Hello? Okay, fine. So we'll start the session now. So, just a minute, I think people are joining. Uh, yes, uh, so someone has raised the hand. Jahanera ma'am, Jahanera Momin. Okay, so yeah, you can ask whatever you want to ask. Do we have any question? Okay, fine. So we'll start the session. So, good evening, everybody. First, I'll introduce myself. I am Apurva Soni. I am a PMRF PhD scholar at University of Hyderabad, and will be your TA for this course, RNA Biology. So, I think there are two online discussions, online doubt clearing sessions for this particular course. Am I right? You have two doubt clearing sessions, right? Yeah. So, so can you just anybody can unmute and tell me what all you have learned yesterday's session so that I don't want the things to get overlapped. At the very same time, I want to cover all the topics because you have two sessions. So I want to conduct my sessions in such a way that you get some benefit and there is something different that you can learn in each of the sessions. Yes, anybody who wants to say what just a gist of what you have learned in the last session, like in yesterday's session. Anybody? Anybody want to tell? Okay, then I'll start by in my own way. Just a minute, people are joining. Okay, so I think now some people have joined just now. I think they will be joining now. Fine. Okay, so we'll start the session. So in today's doubt clearing session, we will be covering the topics that have been covered in week 1 as well as in week 2. So, my way of doing with the live sessions is that first I try to revise the portion that has been covered and then we'll solve some questions. So, some questions would be new, and some questions might have some repetitions of what you did yesterday. So, that would be just a revision. And also, if you have any doubts, some points that you could not understand, you can always ask me. Okay, so this course is RNA biology. So, this course is all about the evolution of RNA and how RNA evolved. So, why RNA was used as a genetic material? What were the disadvantages that RNA could not continue as a genetic material? And then, what led to a shift of genetic material from RNA to DNA? Then we'll be seeing that what advantages does RNA had over other biological molecules because of which RNA actually existed. And then eventually we'll try to move and see the various molecular biology processes that occurs in a living organism such as transcription, translation, RNA processing, splicing, etc. Okay, so we'll be going with the PDF also. So if you must have gone through the PDF and the live lectures. If not, you can see that this is the course outline. Um, so we have a lot of things to cover here. Just a minute, students are still joining, so I think for the first few minutes it would be a little chaotic. Okay. So if you have any questions, please unmute and ask, okay? Because I might not be able to see the messages all the time because I'm changing the screen. I'm not on this screen. So you can just unmute yourself and ask your questions if you have a doubt. Fine. So in this particular course, we first will start with the evolution of RNA, that how RNA evolved. Okay, so when there was life on earth, how life started, how RNA evolved, what is the RNA world hypothesis, that will be see. Then important property of RNA is that RNA can act as a catalyst. That means it can self catalyze itself, and it can also helps in the catalysis of other biological reactions, for example ribozymes. Then we'll be seeing the life cycle of RNA, that is transcriptional, export, translation, etc. Then structure, function, and regulation of RNA protein machinery, ribosome, spliceosome, telomerase and telomerase capping, function of small RNAs, that means microRNAs, then RNA interference, which is very important topic, then some non-coding RNA, heterochromatin and X inactivation, and overview of the techniques used for RNA research. So this will completely summarize most of the topics which are related to RNA. So I think some students are still joining. Just a minute. Yeah. So can you just quickly tell me that you all are like bachelor student or master student? Just can can you tell me? I want this session to be interactive. Okay, this is a doubt clearing session, not like a regular session where you are just going through the pre-recorded lectures and you have to just listen it. You have to interact. Okay, then only it would be fun, and then only you will be able to see for whole 2 hours. Yes, you can say. Anybody?
Masters, ma'am.
Master students. Okay, fine. And so all are master student, or some are bachelors as well? MSc students are there. Fine. So that means if you are master students, then you have some little bit idea about RNA. So what you can understand from RNA? Can anybody tell me anything, any property, not just like a definition, but what you know about RNA? I want one answer from anybody of you. Any answer? What is RNA according to you? You have studied yesterday also, you had your one live class. So what do you think about RNA? What is RNA? What are its property? Anything, any point about RNA? Tell me. You can type if you are not comfortable in speaking. You can type also. I'm still on this screen. You can type. But I want some answer. Yes, Jahanara, you are raising hand. You can say whatever you want to say, or you can just message. So nobody is answering anything. Nobody knows anything about. Yeah. Someone has said sonal. Okay. Precursor for the synthesis of proteins. Okay. So basically, you are talking about that RNA, which is used for the, I mean, from the DNA, it is converted into RNA, and then that RNA is used for the synthesis of proteins by the process of translation. That is your thing, correct? Then you said that believed to be the origin of life. Okay. And Jahanara has said it is genetic information. Okay, so it carries the genetic information. Fine. So you have a little bit basic idea about what RNA is. Okay, so this phrase RNA was coined by Walter Gilbert in 1986. Okay, and he said that self-replicating RNAs were precursor to current life based on DNA, RNA, and proteins. Okay, that means when life originated on Earth, then there was not no such thing like DNAs. It was RNA that was the first genetic material that was used, but because it has certain properties because of which it cannot be used for a longer period of time, because it was very unstable, it was very much prone to mutations, and therefore eventually the genetic material got shifted from RNA to DNA. Okay, so then in the first this thing lecture, we have studied about, I'll just give a gist of what important points we have studied in our lecture 1. So in a lecture 1, we have studied about the beginning. That means how the origin of life started, and from the origin of life, how the concept of RNA actually started. So what they are saying, that self-replicating RNAs were precursor to current life based on DNA, RNA, and protein. Okay, and then there were several theories on how life originated and how life evolved. There were very much versions of that, and then comes the theory of RNA world. What does that theory say? It says that RNA can perform both genetic and catalytic role. Okay, so that is actually a very important property of RNA, that RNA can perform genetic roles also, and it can perform catalytic roles also. So when I talk about genetic role, what is the meaning of genetic role? That means it can carry the genetic information from one generation to another. That is the genetic role. And catalytic role means, I have written in brackets also, self replicating. So, what is the meaning of self replicating here? That means, are you able to hear me? Is my voice audible?
Yes, ma'am. Yes,
Okay,
ma'am.
fine. So I think other students have also answered. RNA is a self-replicating molecule, answered by Sweetlin. Then Janvi has answered RNA is used for the synthesis of protein. Good. So I like this. So I want people to interact. If you're not comfortable in speaking, you can message me, but always respond. That is what I want in my class. Okay. So catalytic means what? Catalytic means that RNA does not require any enzyme for its replication. Fine. So RNA has its own RNA dependent RNA polymerase. And RNA does not require the assistance of other enzymes, other molecules for its replication. Unlike DNA cannot replicate by its own. It needs the assistance of other enzymes. It needs the assistance of other molecules. But RNA, so initially RNA started as a genetic material because it can self-replicate. It is a very important property of RNA. So then in the first lecture of week 1, we started with the basics of what is life, then the various versions of life. So important point to see over here is, for your examination point of view, is that anything which has life is something which has the ability to reproduce. If something is not able to reproduce, then it is not considered as a live thing. For example, I don't know how many of you have done some kind of mammalian or animal cell culture. But when you do animal cell culture or mammalian cell culture, when you culture your cells in vitro, then if your cells are live, then they will divide and they will replicate, and then your flask, in whatever flask you are growing your cells, say T25, T75, whatever flask, it will become full of cells, confluent. You're plating your bacteria on agar. If they are live, then you will see colonies. So what is happening? They are able to reproduce themselves. They are able to replicate. So that means they are live. Okay. So anything which has life will be replicating, will be able to reproduce. When we are saying in terms of mammals, they are able to reproduce or they are replicating. So they are just producing their copies. That is important. Okay. Life means they should produce their copies. Fine. Then becomes, this is the first criteria of something to be considered as life. Second criteria is the ability to acquire particular molecules and use them in controlled chemical reaction that maintain growth suitable for life and contribute to growth. That means you're you're able to grow, you're able to survive. Then there are other requirements also, such as you require some kind of membrane, like a plasma membrane, so that can precisely control the in and out of specific substances. So these are all basically, in a gist, the requirements for any particular thing to be considered as a live thing. Okay, so we are just dealing, in the first week we just dealt with the basics of what life is, what are the theories of life, how we can say a particular thing is live or not, and then we eventually comes to the RNA world. Okay, so the RNA world, so this RNA which can perform both genetic as well as catalytic function. Okay, then RNA world was of self-replicating ribozymes. So in this week 1 and week 2 class, this ribozyme is a very important topic for you. Okay. So questions can come for your exams on this ribozyme. So ribozyme is important. You will be studying, or if you have already studied, there are so many questions and so many topics related to ribozymes that you have to see. Finally, so there were uh, there were some theories which were related to RNA, and there were some evidences to support this theory. What was those evidences? That ribozymes. So ribozymes, basically ribonucleoprotein, which are self-replicating, so which kind of again reinstated the fact that RNA is there, and RNA is actually a very predominant thing than ribosomes. So ribosomes, basically the active site which is responsible for the peptide bond formation is actually an RNA. Okay, so the peptidal transfer is activity. So in a ribosome, you have multiple sites. You must have studied EPA site, and then peptide formation takes place. So that bond formation is catalyzed with the help of an RNA. So this ribosome and ribozymes both cumulatively reinstated the fact that yes, RNA is very important, and there is an RNA world which is very much responsible and contributing factor in the origin of life. These were just some of the milestone. I think this is nothing to explain over here. And then yes, there was this you can go through it again, that there is a kind of a meteoroid to which uh, uh, so whatever components which were there in this meteoroid, they kind of resembled the components which were present in the origin of life. Nothing much to explain in this. So up till now, so this was just a brief gist about the lecture 1 of the week 1, where we have studied about the RNA world, the versions that RNA can perform both genetic and catalytic function, and some milestones in origin of life sciences. There is nothing to explain very much. Then comes very important, this experiment, Miller-Urey experiment. Now this experiment is important not just for your this NPTEL examination point of view, it is also important for some of your competitive examination point of view. So I just have a small quick question to the students over here. So how many of you are interested in pursuing higher research, like going for PhD? You can raise your hands if you are interested in going for PhD after MSc or any kind of research. Are you interested, like anybody of you interested in going for PhD afterwards? Quick question. Okay, there is one student who is interested. Only one amongst 37? Two. So there are some students who are interested. Only two. Two out of 37. Three are there. Okay. Okay. So someone is pursuing PhD. Okay, that's really good. Okay. So that means you have a direction towards inclination towards research. So for undergoing any kind of research, you have to have clear some competitive examinations like CSIR or gate also. I think gate, you might have gate as of now, but yes, you need CSIR, DBT, ICMR, some of the examinations. Okay, so I will try in this particular course to, like, whatever we will be studying about RNA biology. So I will try to point and tell you some important topics that will be helpful for your further competitive examination point of view as well. Okay. So while we are reading about the things, so I'll tell you that these points are important for your exam purpose also. So this will benefit you not only for your this examination, your NPTEL examination, but also for your some other competitive examinations. So this Miller-Urey experiment is important. Okay. So why this experiment is important? So basically, this experiment was conducted to to demonstrate how life was originated in early Earth. So basically, they had two flasks. You can see there are two glass flasks which were connected with tubes, glass tubes. Okay. So Then in this flask there was boiling water. Okay, this is the lower chamber and this is the upper chamber. So this lower chamber, it represents oceans of the early life, and this upper chamber, it represents the atmosphere of the early life. So basically they tried to replicate, they tried, Miller and Urey, they tried to replicate the conditions, they tried to replicate the conditions of that particular era when life originated. So this experiment was conducted in 1953. So what they did was that when this water is boiling, so which is actually demonstrating the boiling water of the oceans, okay, then there are water vapors, and then here upper chamber, when this water, the water vapors, it reaches the upper chamber, that means the atmosphere, there is electrical discharge. So for this electrical discharge, they have put electrodes, because that time life was originating, so there were a lot of electrical discharge, extensive heat, everything was there in the early atmosphere. And because of this electrical discharge and this water vapor, there was formation of gases. Okay, and these were the initial gases which were formed, and then these gases, when they condensed into water droplets, it led to the formation of organic compounds, which you can see over here. Okay, so that means boiling water, when it is condensed, and then when this boiling water goes into this particular second chamber, where there is electrical discharge also happening because of the electrode, the formation of these molecules takes place. This represents the way how life must have been originated at early times. Okay, so that is what they are saying, that volcano and hydrothermal vents generated extensive heat, which provided a catalyst for biological reaction to happen. Okay, and then the evidence for this was the rRNA phylogenetic tree has hyperthermophilic organisms clustered near the base of archaeal and bacterial origins. What is the meaning of this? Let us see in this particular lecture 2. Just a minute. Yeah. So you can see that this is how the experiment was conducted. Okay. And when they compared the amino acids which were formed in this experiment uh, with the one of this Murchison's meteoroid, you can see the similarity in the amino acids which were formed in the meteoroid and in the experiment. You can see the similarity that was shown to you. Fine. So basically, this is how the experiment that has been conducted. This is how it must have happened in the real life. That because of so much of hydrothermal habitats and volcanoes eruptions, so much of heat is generating. Okay, and because of that heat it becomes water vapor, and because it was an early atmosphere, there was a lot of striking electrical discharge, which created and particular which led to the formation of all the initial molecules. And then eventually when they settled down, they combined and they formed uh, whatever molecules, like you can see over here. Yeah, then they form the initial water, carbon dioxide, sulfur dioxide, everything. Okay, now how we can say that this actually happened? Like if I am saying, if the facts say that earlier atmosphere was very high temperature, it was a very high temperature environment at early times. How we can say? We can say because when you go through the rRNA phylogenetic ribosomal RNA phylogenetic analysis, you can see that there are certain organisms which are hyperthermophilic. These organisms are hyperthermophilic. What does it tell? That these organisms, first thing that it tells is that these organisms are related to each other. Second thing, that the earlier atmosphere was very, very high temperature atmosphere. That is why these organisms that were present in the early atmosphere, they have adapted themselves in a way that they can survive at high temperature. Okay. So this is basically this rRNA phylogenetic analysis. It tells that our ancient time was a very high temperature environment because of these hyperthermophiles that existed and they still exist. Now I have a question that why you think this rRNA analysis can help in this? Because we are studying about RNA. This all theory is okay, fine. But how rRNA can tell us about the phylogenetic analysis? Can anybody tell me how rRNA is helpful in telling about the phylogeny? How it can help in phylogenetic analysis? We are studying about RNA here, right? Does anybody know that ribosomal RNA, ribosomal RNA is used for phylogenetic analysis? Tejaswini, you want to say something? Because rRNA is conserved. Your voice is so Khushbu, you are telling right. Tejaswini, your voice is not clear. I think there is some network issue. Do you want to say something? Fine. So yeah, so Khushbu Negi, you are somewhere right. So genes for ribosomal RNA are highly conserved in many different species due to their slow rate of undergoing mutations. You can note it down. It's an important point. Why I'm saying, because it is important for your DBT exams point of view also. Okay, they ask questions about RNA, uh, ribosomal RNA phylogenetic analysis. So basically, ribosomal RNA, they undergo very less mutation. That means the mutation rate of ribosomal RNA is very slow. And because the mutation rate is very slow, that is why they are highly conserved in many different species. And that is why they can be used as markers or for the phylogenetic analysis. Okay. I'll request Tejaswini to please mute your mic. If you don't have anything to say, you can mute it. Tejaswini, can you mute yourself? Fine. Yeah, thank you. Okay. So finally, we can say that RNA world exists because RNA can store genetic information. Okay. RNA can be a temporary copy of genetic information. Fine. RNA can be a carrier molecule. RNA can be working part of cellular machinery, and RNA can work as a catalyst. For example, ribozymes. Okay. So this is what we have learned in the first two lectures. Then okay. So other things are that RNA, what are the subunits of RNA? So the subunits of RNA is a phosphate group, ribose group, and base like adenine. Okay. So phosphate was formed because of the rock weathering processes, and how ribose and base formed, you have must have seen in the subsequent lectures. We'll get into detail one by one. Okay. So then there were some evolution theories, not much to discuss. You can read about them. Okay. So fine. Now we'll go through some questions. So what I'll be doing, that I'll be just revising like one or two lectures, and then we'll go through some questions, and then we'll revise some lectures, and then we'll go through some questions. Okay. So like this, what will happen, that you will have a revision for your assignments and for your exams also, and then you will have some questions, this thing also. So some questions might be repetitions because we are bound to take them. That is our format, but I have also included some fresh questions for you. And in subsequent classes also, I'll be including more new questions. So the first question is, RNA contains repeating units of A, deoxyribonucleotides, ribonucleotides, deoxyribonucleosides, ribonucleosides. So what is the correct answer?
Ribonucleotides, ma'am.
Okay, fine. Correct. Ribonucleotides, you are saying, right? Okay, okay. So many students have answered correctly. The answer is ribonucleotides. Fine. So when I'm talking about tides, that means phosphate group is there. Sides means phosphate group is not there. And deoxy means it is a DNA. Okay, because at the two carbon instead of OH only H is there. So that is your DNA. Correct answer. Good. Second question is molecules which play the key role in transfer of genetic information during protein synthesis are DNA, RNA, nucleic acids, lipids. What is the correct answer? RNA. Correct answer is RNA. So basically the question is saying that in the transfer of genetic information during protein synthesis. So during protein synthesis, actually from DNA it forms RNA and then it forms protein. So basically RNA is the intermediate which play a role during protein synthesis. So correct answer is RNA. Fine. Next question is which of the following statements is correct regarding DNA and RNA? Okay. You have to select the correct statement from this. DNA is highly reactive. RNA is not catalytic. RNA cannot be easily degraded. DNA is a better genetic material than RNA. Which is the correct answer from this question? In this question, D. So students are saying D. So D is DNA is a better genetic material than RNA. Why? Can you tell me why DNA is considered a better genetic material than RNA? You can type why DNA is considered more stable than RNA. Yes, correct. Why DNA is more stable than RNA? Again, one question. Yes. So Prema, Sonal, Khushbu, everyone is right. DNA is more stable than RNA. So why you think DNA is more stable? Yes. So correct. So students are fine. Absolutely correct. Good. I'm liking that students are responding. So DNA is double stranded. So one reason that DNA is more stable is because DNA is double stranded. Also, it is less prone to degradation because it is double stranded. Also, there is 2 prime H. At the second carbon, there is H. In RNA, there is OH. So that also uh, contributes in the stability of DNA. Okay. Next question is at what rate does the RNA mutate as compared to DNA? A. Faster rate. So here the correct answer is D. Now, A is faster rate, B slower rate, C moderate rate, and D depending on the medium. What is the correct answer? At what rate does the RNA mutate as compared to DNA? So students are answering A. Let me see what is A. Faster rate. Yes. So RNA mutates at a faster rate. And that is why again RNA is not as much stable as DNA. So can anybody tell me why RNA mutate at a faster rate than DNA? What is the reason why RNA mutates at a faster rate than DNA? Yes, correct. So Sonal is telling correctly that it is single stranded. Okay, so it is much more prone to degradation. Okay, whereas in DNA, what happens that there is hydrogen bonding. So nitrogenous bases are already occupied with each other in bonded form. But in RNA, there is only one strand. Mostly RNA occurs at single strand. So nitrogen bases are open for bonding. So that is why uh, it is less stable. Also because RNA has its own catalytic activity. Okay. So because RNA has its own catalytic activity, It does not need any enzyme or something. That is why uh, it can cleave itself, it can rejoin itself into some other place, and therefore it can mutate and change its version. uh, so the best example to study about how RNA can mutate at such a faster rate is COVID, that we all have suffered since last 2.5 years or 3 years. So why there were so many variants of COVID virus? Because its genetic material is RNA, and RNA is prone to mutations. It can mutate very easily. And now you know why it can mutate very easily, because it is single stranded, it has self catalytic activity, so it can cut itself at some place, it can join itself, it can transpose at other places. So all these are the reasons why dealing with virus is very difficult, because especially when it has an RNA genome, because then it becomes very prone to mutations and it can change. So we cannot have just one vaccine. If we have one vaccine for against one strain, then RNA can change itself, it can mutate, and then we don't have vaccine for the strain. So that is why it becomes so difficult to combat diseases which are viral diseases. Okay, fine. Next question is, RNA codes are used for translation into which of the following? Very easy question. Genes, DNA, proteins, carbohydrates. What is the correct answer? C, proteins. Correct? Yes. So everybody has answered it correctly. The correct answer is C. So these are actually very simple questions. I think these questions even does not require any class to be uh, attend. But okay, so this is our first class. So we'll eventually, as our course content will increase, we'll also increase the difficulty level of questions, right? So I will have questions which are unique, and I have to take questions which you might have studied already in your Monday's online class. But don't get disheartened, because we will have ample amount of questions. I have to take those questions because that's compulsory for me. That's why I have to take those questions. But I will try to have more and more new questions for you, okay, so that you can get more practice. Okay. So now we dealt some questions. Now we'll come to the lecture 3 of week 1, which talked about origin of monomers. Okay, so in this we have learned about the ribozymes and ribosomes, and the further evidences to reinstate the fact that RNA actually existed, like ATP, SAM, biotin, coenzyme, they all are nothing but RNA. Then how the formation of sugar take place, matlab how the formation of base, nitrogenous bases, and sugar, and how the polymerization of nucleotide take place, and then ultimately the Chargaff rules. So these are the points that we have covered in lecture 3. So let's just quickly go to the PDF and revise it. Okay. So basically, further evidences to reinstate that RNA world existed is because these cofactors have RNA fragments attached to them. Okay, so such as ATP, S adenosylmethionine, flavin adenine dinucleotide, then nicotinamide adenine dinucleotide, coenzyme A, and biotin. So these all have RNA as their cofactors, which further reinstated the fact that RNA existed. It has role in multitude array of processes. Now, now comes that how the formation of an RNA takes place. Here we are talking about formation of RNA molecule. Basics. So you have ribose, sugar, you have a nitrogenous base. When they do these two things combine, you have a nucleoside. When I talk about nucleoside, that means it does not have the phosphate group. Then phosphorylation takes place, and when phosphorylation takes place, then you have activated nucleotides. That means here phosphate group is present. So you should always remember that in such metabolic or cellular processes, When you add phosphate group, then that particular molecule actually becomes activated. So in many kind of uh, cellular pathways or metabolic pathways, the addition of nucleotide, the addition of phosphate is required for the activation of the molecule. Okay, so when you add phosphate, then your particular thing becomes complete RNA. And then when polymerization takes place, then you have a RNA polymer with you. Basics we all know about all these things. It's a very basic things. Now in the prebiotic era, did we have things for the synthesis of RNA, that means sugar and bases? Yes. The question here comes that how RNA was formed in the prebiotic surroundings. This is the question. How RNA emerged? So did they have some uh, starting material? So yes, starting materials were there. So what were those starting materials? How RNA formed? So you can see, just a minute. Hmm. So सबसे पहले first thing is the formation of sugar. So the sugar formation occurs with the help of one reaction which is known as Formose reaction. So you just need to learn that how this might be an important question, that how the synthesis of sugar occurs, or synthesis of sugar is supported by which reaction. So the reaction is Formose reaction. Okay, this reaction is required for the synthesis of sugar. Okay, so then there is a whole detailed description about Formose reaction. You can read about it. Main thing is that you should understand that in the prebiotic era, when there was not much things available, then how this sugar actually started. So you have formaldehyde. So two molecules of formaldehyde, when they condense, then they form glycoaldehyde, and then through various reaction, the formation of sugar takes place. So what they are writing, that glycoaldehyde reacts to form a mixture of tetrose, pentose, and hexose sugars. So by with the help of Formose reaction, which involved the initial substrate as formaldehyde, because initial substance is formaldehyde, therefore reaction is known as Formose reaction. Correct? Now this is how sugar synthesis took place in the prebiotic era. Okay, and then your uh, mm, phosphate phosphate, because of the rock weathering, the rock weathering led to the formation of phosphate. And uh, yeah, then how the nucleotides polymerize. This is important. So the polymerization, that means bond between the two RNA molecules. That bond is known as a phosphodiester bond. Important. Okay. The bond between two RNA molecules is a phosphodiester bond. It is basically a condensation reaction in which a water molecule is removed, because there is a bond formation between the phosphate group of one nucleotide and the OH group of another nucleotide. Okay, so which leads to the formation of water molecule. This water molecule is released, and then your phosphodiester linkage is formed. Okay, then this is just the DNA phosphate backbone RNA. You can just go through it. These are all the reactions. You just have to, need not to just mug up everything. You need to just know the basics and the concept. Then we come to the synthesis of nitrogenous bases. So for the purines, I think this question was solved in yesterday's class also, that the synthesis of purine takes place from HCl. Okay. Then this is the mechanism you must have studied yesterday in yesterday's class. Then the basic structure of purines and pyrimidines that we all have been studying, and Chargaff's theory. So can anybody tell me what is Chargaff's rule? Chargaff's rule? What is
Purines
Chargaff
are
rule?
equal. Amount of purines equal to pyrimidines.
Okay. So, this Chargaff rule is very important. Why it is important? Because a lot of questions from Chargaff rules comes in your GATE and DBT examinations. Okay. So, if you want, I can give you like in if it all depends on how much interest you people will show. So, there are some important questions that I can share with you, sample questions one or two in the subsequent class if you want, where we can see what kind of questions comes in competitive examinations. So they will give you that in a DNA molecule A is present 50%, then what is how much T is present, or how much percentage of G is there, how much percentage of C is there. So like these questions ask. Okay, so a lot of people have answered in the messages also. Okay, so Sonal has said A is equals to T, G is equals to C. Okay, G is equals to C. Okay, so everyone has said correctly only. So if I write something, I'm writing something, you tell me it is correct or not. If I say A is equals to G, is it correct? Is it correct? Yes or no? False. Yeah. Why it is
No,
false?
ma'am.
No, correct. So A is not equals to G, A is equals to T, and G is equals to C. Okay, so A is equals to T means that much A, that much T has to be present, because all the A will bond to all the T. So how many hydrogen bonds between A and T?
Two, ma'am.
Sorry,
Two.
two. Correct.
Two, ma'am.
And between G and C?
Three. Three, ma'am.
Three. Correct. So suppose if I have a
Three, ma'am.
sequence, okay, and in my sequence, suppose my sequence is, I am writing in the message, okay. This is a sequence. So does anybody know how to calculate the melting temperature of this particular sequence? It's not related. I'm just asking some question. It's basics. Does anybody know if I want to know
Based
that?
on GC content, ma'am.
Yes, based on the GC content. Okay, that is fine. So, do you know this formula? 4 into G plus C plus 2 into A plus T? Does anybody know this formula? Okay, so I think we'll subsequently I'll tell you about this formula also. So I just want to tell you something, that if in a particular strand, this is a DNA strand, okay, this is not an RNA strand because it is T here. If I have to write an RNA strand, I have to write U. Okay, so if this particular strand will have a higher melting temperature or a lower melting temperature will depend upon the content of GC. Because if GC is more, that melting temperature would be high. Why? Because to break one GC bond, uh, for one GC you have to break 3 hydrogen bonds, and to break one AT you have to break only 2 hydrogen bonds. That means if GC is present more, melting temperature would be high. Just one basic uh, topic out of the context, not exactly, but I just wanted to tell you. Fine. So these are the things that we have studied in this particular lecture 3, Chargaff rule. It is important, but I think students know. So basically A is equals to T and G is equals to T. So what are the purine bases? What are the bases which comes under purine? Tell me.
A and G. Adenine, guanine.
Yeah. So adenine and guanine, they come under purines, and pyrimidines.
Thymine, cytosine, and
Cytosine,
racil.
hydroxyl, and adenine and cytosine.
Yes. So these are the purine bases and these are the pyrimidine bases. So in what other ways we can say Chargaff rule? Are there any other ways also to write Chargaff's rule? in context of purines and pyrimidines? Tell me. So basic, I think, yeah. So I think Khushbu has written correctly. Okay. The sum of purines is equals to sum of pyrimidines. Yes. So basically, you know it correctly that the sum that purines, uh, okay, purines, basically purines is equals to pyrimidine because purine will pair with pyrimidine only. Purine will not pair with purine. You can see over here, purine is A and pyrimidine is T. So purine will pair with pyrimidine, and here also purine is pairing with pyrimidine. Purine pairs with pyrimidine. That means, like, purines should be equals to pyrimidine, then only it can form perfectly pair. Another very important point to understand about Chargaff's rule is that this particular rule is applied only when it is a, what is it? If I say there is a DNA molecule which is single stranded, can I apply Chargaff's rule there? Tell me. In a single stranded DNA molecule, can I apply
No,
Chargaff's
ma'am.
rule? No. Yes.
No,
So,
ma'am.
so your DNA has to be double stranded,
No,
only
ma'am.
then you can apply Chargaff's rule. It is important. Okay. I think it is enough for the Chargaff rule. Now we'll just move to our next topic. So then in lecture 4, we have studied about the shift of the genetic material from RNA to DNA. Okay. Now their DNA is very stable. So that means for to carry out any catalytic reaction, DNA is very stable. That means it cannot catalyze by its own. It needs some enzymes from outside. Then DNA is too simple and too stable to act as a template. Then why did this genetic material move from RNA to DNA? DNA does not have any self-reactive mechanism also. DNA is too simple, it is so stable that for acting as a template, it needs so much of energy also. Then why there was a shift of genetic material from RNA to DNA? Why there was a shift? Can anybody tell? Despite having these two disadvantages, even then, the genetic material today in today's world is DNA and not RNA. Why? Any reason you can think of? Okay, I'll tell you one very basic reason that RNA, okay, RNA has self catalytic mechanism. Yes, it does not need any template for its synthesis. Okay, it can self catalyze itself fine. But there is one problem with RNA. And what is that problem? That RNA is very reactive. That this self-reactive, self-catalytic property that RNA has, it's actually inhibiting it from being a genetic material. That is, like, this property is the only reason which is why RNA is not a genetic material. Because RNA is so reactive that it is not stable. It is easily prone to degradation. Okay, RNA can be degraded very easily. So, our genetic material, which has to move from one generation to another, it has to be stable so that it can pass from one generation to another. And it should not mutate in such a high rate that it is not able to transfer the important information from one generation to another. Okay, for example, yes, Jyoti, you are right. It has more oxygen and it has, okay, it has a hydroxyl group which is rendering it more reactive. Fine. So suppose uh, our genetic information, suppose of having 5 fingers. Okay. So this 5 fingers genetic information is present in a gene X, let's say. It's a very random example that I am giving you just to show that how this mutation can create problems. There is a gene X which is responsible for 5 fingers. Okay, or you can say that for dividing the 5 fingers. Sorry, my drawing is not good. So if this gene X is not present, then there would be some mutations, and then what will happen? Either you will have less fingers or more fingers, or some kind of uh, deformed shape of hand would be there. So now this gene X has to be correctly transferred from the parent to the offspring. Now suppose if this gene X is sitting on this RNA molecule. Okay, this gene X is sitting on this RNA molecule, and because this RNA is so reactive, it has so much of catalytic activity, that because of this own self-reactive activity, it has cut out the half portion of this gene. Because this gene was present, suppose this was the region where gene was present, and half of the region of the RNA got spliced out. Now you have only half of the gene. So what will happen? In the offspring there would be deformity. The offspring which is produced will not have the correct 5 finger hand. It will have some kind of abnormality. So that means this RNA is not a good vehicle to carry this gene from one generation to another. On the other hand, if this gene is present on DNA, because it is so stable, it can easily pass this gene from one of parent to the offspring. Is it clear to everybody what I'm trying to say? Suppose I want to go from my home to my college. So I have to choose a vehicle. So I have to choose a vehicle that is that is fast enough, but I cannot compromise with my safety. For me, safety is important. Even if I'm reaching 10 minutes late, it is fine, but I cannot compromise with my safety. Same principle applies with RNA and DNA. That RNA can be fast, but RNA is not safe. And why RNA is not safe? Because because proofreading activity. You know about this activity, proofreading activity? So this proofreading activity does not occurs in RNA, whereas this activity is present in DNA. What is proofreading activity? Can anybody tell me? Does anybody has any idea about this proofreading activity?
It removes mismatched extras from DNA.
Sorry, Jyoti, can you repeat? I'm unable to listen. I think there's some problem in the network. You can type. What is this proofreading activity? So basically, I think Jyoti was telling correct only, but because of some network issues, I could not hear her properly. So, so when DNA replication is taking place, okay. So after the replication, just like you proofread your assignment before submitting it to your professor or your teacher, you proofread it. That means you're just rereading it to see if you have not done any mistakes. Or if you're writing a mail to your professor, to your supervisor, you just reread it to see that there aren't any mistakes. That is proofreading. So likewise, what DNA does is that during replication, there is a proofreading activity that sees that are there any mutations that has occurred during the replication process. Suppose if it has the in DNA it has to incorporate A, and by mistake it has incorporated C. So so during proofreading activity it will see that acha, here C is incorporated instead of A, so it will correct it. And if it is not able to correct, then it will halt the replication at all. So in this way, DNA is much more safer. Whereas when RNA, during RNA transcription or during RNA synthesis, what happens that there is no proofreading activity that takes place. And therefore, yes, remove mismatched bases from DNA. Correct. Jyoti, you are correct. Bhavya, checking the error or correction, absolutely correct. Swarna, corrections. Okay, everybody is saying the correct thing. That corrections, whatever corrections have to be done, that does not happens in RNA. And therefore, RNA, whatever mutations that has happened, it just keeps on going and it increases. And so I don't know, I think you must have some idea about it, is that the DNA repair mechanisms that corrects the mutations or some problems that has occurred in the DNA. So that is one of the reasons that if these mutations are not corrected at DNA level, because of some DNA repair or repair is not happening proper, cancer formation takes place. Okay, because what happens, that if that incorrected DNA goes exit from the cell cycle and it replicates, then there is a mutation, and then mutations can ultimately lead to cancer. So that is why in our body has made a very beautiful system where it tries to uh, it tries its best to reduce the chances of any kind of mismatches or any kind of errors. Our body has made it very beautifully, but because of some external oncogenes or signals, if some mutations happens and DNA is not able to correct them, then it leads to the formation of cancer. Anyways, so what I was trying to tell you, that because RNA is much more reactive and RNA has much more mutation chances, therefore RNA cannot be used as a genetic material, and hence DNA emerged as genetic material, and there was a shift of genetic material from RNA to DNA. Is it clear to everybody why there were why the genetic material changed from RNA to DNA? Because RNA is very unstable. Okay, so this was basically what we have covered in lecture 4. Quickly, I just want to cover one thing more in lecture 4, and that was hmm. So okay, so RNA takes the lead. So, see, what we have studied about how DNA uh, emerged as a genetic material, that is one story. And another story is that when the in the prebiotic era there were so many different molecules present, but why RNA only took lead at that time and becomes genetic material? Because it was able to replicate and it was able to catalyze. Okay, because it was able to replicate itself, it was able to replicate itself, and it was able to catalyze. Therefore, at that time in the prebiotic soup, RNA took the lead and become the genetic material. But eventually the shift came and DNA become the genetic material. Okay, this is what evolution is. First something, then other thing. So you just evolve and become more and more useful for your own body. Okay, so Tejaswini and Jahanara, you have raised your hands. So one by one, you can ask questions. Tejaswini, what is your question? You can ask. What is your question? Tejaswini, what is your question? Jahanara, your question. I'm sorry if I'm not correcting your name properly. Okay, fine. Then we'll do some questions now. First, we'll do some questions and then we'll see what was there in lecture 5 and 6. Okay, so question is, name the RNA molecule which takes part in the formation of ribosome. mRNA, tRNA, rRNA, or gRNA. What is gRNA? First, tell me what is the answer of this question. RNA molecule which takes part in the formation of ribosome. Yes, rRNA is the correct answer. So the molecule which takes part in the formation of ribosome is rRNA. Now tell me what is gRNA. Can anybody tell me what is gRNA? Guide RNA. Exactly. So what is the use of this guide RNA? Where we use this guide RNA? Any application? Latest application of guide RNA use? Anybody knows? Okay, I'll give you a hint. That application, whoever made this application got a Nobel Prize also. CRISPR. Exactly. So this guide RNA, so can you just spell your name? I'm sorry. Harin Narayan has correctly answered. It is CRISPR. I'm sorry, I'm extremely sorry if I'm unable to pronounce your name correctly. But yes, so CRISPR is the latest technology where we use guide RNA. So basically, guide RNA is used in CRISPR-Cas technology, okay, for generating knockout or knock-in species, and many more applications are there. Various applications are there. So yes, RNA DNA targeting enzymes. Correct. Meghna, you are correct. So, knockout means to knock a gene out, to remove a gene out of the system. Knock in means to put a gene in, to give a gene into the system. So, here we are deleting a gene and just knocking it out of the cell. Here we are giving externally one gene into the cell. So, in both the cases, we can use guide RNA with the help of CRISPR-Cas9 technology. So in my PhD work also, I use this CRISPR-Cas9 technology. So I use this guide RNA for some work, for some of my objectives. Fine. Next question is, which of the following criteria need not to be fulfilled by the genetic material? Okay, that means which of these criteria is not compulsory for a particular molecule to be called as a genetic material. So the first is occurrence of replication, should be able to express itself in the form of both characters, provision of scope for the mutation that is required for evolution, it should be chemically and structurally stable. Which criteria is not important? Which, what, tell me the answer. What should be the answer? Okay, someone make nice saying B. I'll just open this so you can see the options and tell me. Occurrence not important. Read the question. I'm talking about not important. What is the answer? B. Correct. So many students have answered it correctly. So any particular molecule to be a genetic material, it should definitely replicate itself. Yes. It should be stable, of course. Provision of scope for the mutation that is required for evolution. Now, see, here comes, some people might get confused. This is in this, it is very much um, clear that B would be the answer, because it from nowhere it is near to the criteria that we know or studied. But some students, you can ask that I am saying that RNA does not is not considered a genetic material now because it is very much prone to mutation, and now here I am saying that it should have mutation. So what should be your logic behind it? Here they are saying that there should be some scope for evolution, so there should be some mutation, and then I say that because RNA, the mutation rate was so high that it could not sustain as a genetic material. So these two are very contrary statements. Then what do you think? What should be the logic? Try to think an answer. Try to think an answer. Can anybody suggest any logic behind this? Some mutations required for adaptation. Correct. So Sonal is saying absolutely correct. So that some mutations are required for adaptation. So, one thing that I really want you all to understand is that whenever we talk about mutations, we think of it as a villain. We think of mutations as something very bad. Okay. But in reality, 90% of the mutations that occur in our body does not lead to any kind of disease or harm or cancer or anything big. Okay, either they are reverted back because of the DNA replicating machinery, or they does not show any phenotypic characteristic. Okay, they may be nonsense mutations. You must have studied about missense, nonsense, and all those different types of mutations. So in order for a particular organism to evolve and to adapt with its environment, it has to incorporate certain changes in its body, in its lifestyle. So just like when we go to a different place, when you must have started your college, So for many students, for first few days or first few months would have been a little difficult. You took time to adapt to the new environment, and eventually you adapted, and now you have friends and you're very happy with your environment. But initially you took some time to adapt. So then, but then you adapted. So adapted means you changed your ways in a certain way. So similarly, our body need to change its ways in the form of its expression of genes in certain ways to adapt with the changing environment. That means our body need to have certain kinds of mutations. But as it is said, everything in excess is bad for our health also, and on at a molecular level also. Okay, too much proliferation of cells can lead to cancer, although proliferation is required for maintaining a, a proper healthy cells, but too much proliferation, it can lead to cancer. In a similar way, mutations are required for evolution, but not hypermutative genetic material is good. That means mutation should happen, but in a controlled manner, in a normal manner. It should not happen so much that it is changing entirely the shape and everything. It should happen in a controlled manner. So that is why a genetic material should be able to mutate, but not so much that it is harmful for us. That is what the gist is. Okay, then we come to other questions. So central dogma in biology says that DNA replicates, RNA replicates, protein replicates, or ribosome replicates. So what is the answer of this question? Central dogma in biology says DNA replicates, RNA replicates, protein replicates, or ribosomes replicates. What is the answer? A. Yes. So can any perfectly right? The answer is A. So can anybody tell me the central dogma? How the central dogma works?
DNA to RNA, RNA to protein, ma'am.
Yes, exactly. Devi, you are absolutely correct. So, central dogma says that DNA will replicate. So, DNA to DNA, that is replication. DNA to RNA, that is transcription. And from RNA to protein, it is translation. So, this is what the central dogma is. Sonal is also said correctly, replication, transcription, translation. Yes. Okay. Now, these are true, false. I think some of them you must have done, or some are new. I don't remember now exactly which are repetitive and which are new. Anyways, so the first question is, only bacteria and plant contain ribozymes. True or false? Only bacteria and plant contain ribozymes. True or false? False. Correct. Correct. Everybody is saying correct. Aditya, Hemlata, Sweetilin, Sonal, everybody is saying correct. Good. Then, chemical properties of RNA are different from DNA. True or false? True. Shivani, Swarna, Jyoti, Aditya, Jahanara, Hemlata, Shweta. Everybody is saying Yuva, Ranjini, Trishala, Devi, Prema, Durga. Good. So I am liking. I want this type of class which interact. So it's always good that you interact. So you might have some inhibitions, but I think eventually you will be able to even ask your doubts. And very important thing that I want to tell you, that if you have a doubt related to not just RNA, of course, RNA is our topic that we are studying, so you can always ask all the doubts that you have about RNA. But if in general, if you have other doubts related to any other field, any other topic in molecular biology, or any other topic in biotechnology in general, if I have the answer, then I'll definitely try to answer it. Okay, so you can always come and ask me in the class itself, after the class, anywhere, or you can mail me. But if you have any doubts regarding any of the topics, or anything related to future, as in what you want to do, what are the exams that you can give, what are the scopes and opportunities that you want to have, because not many of, like, even when I was in my masters, I didn't have that good seniors who can help me or guide me of what I should do in my future and where I should go for PhD and all. So this is a very good opportunity for you to clear your doubts in general. Fine. So can you tell me what chemical properties of RNA makes it different from DNA? Okay, RNA is different from DNA chemically. Okay, fine. But what are those properties that make it different from DNA?
Ma'am, RNA contains uracil while DNA contains thymine, ma'am.
Yes, fine. Very good. Jyoti, you are absolutely correct that RNA has uracil, whereas DNA has thymine. And Sonal is saying ribose sugar, again, very correct. So basically, uh, an RNA is single stranded also, but that is not a chemical property. But yes, so these are the things that make it different from DNA. Fine. Next is every RNA has a secondary structure. True or false? Every RNA has a secondary structure. True or false? False. Okay, so students are saying false. Correct. So can you name one secondary structure that is in RNA? So Janvi, Jahanvi, Swarna, Vaishnavi, Shivani, Hemlata, Aditya, Prema. Okay, so you all are saying correct that not every RNA has a secondary structure. But can you name one secondary structure that is found in RNAs? Any one secondary structure that you can think of? Okay, and Vaishnavi is saying tRNA.
tRNA.
Yeah, fine. Then metal ions destabilize ribozyme. Is it correct? Metal ions, they destabilize ribozyme? True or false? False. Jyoti is saying false. Okay. Metal ions destabilize ribozyme. True or false? Jahanara is saying yes. So can you tell me why you are saying yes? What should be the correct answer? True or false? Metal ions, they destabilize or they stabilize? And if true, why? If false, why? Tell me. Okay, so for this, we have to go through, we'll quickly go through the lecture and we'll see. Fifth, we were on fifth lecture, right? Okay. So basically, this is in the fifth. Okay, we're just halting for a few minutes. I have to explain that point that whether metal ions destabilizes or stabilizes. Okay, there are two answers. Jyoti is saying false, and Jahanara is saying yes, that true. So let's find out what is the correct answer. Okay. So first, in lecture 5, we studied about ribozymes, how ribozymes can form um, uh, uh, the secondary structures, and how it can help in the cleavage. So you can see that this is a how it binds. First it binds and then it cleaves and then it dissociate and then it ligates to the other RNA molecule. That is fine. Important points we'll be discussing because we have to go to week 2 also. That question about our metal ions, I want to deal with that question first. Hmm. Okay. So I don't know in which lecture it was, but anyways, so basically ions. So ions are whatever ions we are using, such as in my this thing I told you about metal ions. Yeah. So basically metal ions, they help in the stabilization of the ribozyme. They help in the stabilization of the ribozyme structure and function. Okay. Especially when I talk about metal ions, I'm talking about magnesium ion. Okay. So basically magnesium ions, it helps in the stabilization. So because these metal ions, they can also form coordinate bonds, and not only in the, we're talking about in context of RNA, but if you see, suppose how many of you know about the process of glycolysis? I think everybody knows about the process of glycolysis. Everybody must have read.
Yes, ma'am.
Yes. So, so in okay, so in glycolytic pathway, so when you go from one step to another step, are there any ions which are involved? Mg2 plus, like the cofactors. Have you studied about it?
Yes, ma'am.
So what is their function there? It helps in the proper conversion from one state to another. Okay. So likewise, we can consider it over here also. But still, I'll put this on hold because I have to tell you the exact reason which is given in your notes. So it might come in the subsequent lectures and then I'll tell you. Okay. Next question is, self-splicing introns required the need of a spliceosome. True or false? Self-splicing introns require the need of a spliceosome. True or false? Self-splicing introns require the need of a spliceosome. True or false? Khushbu is saying for, okay, it's false. Yes, yes, correct. It is false. Why you think it is false? Why you think that self-splicing introns does not require spliceosome? Anybody who can tell me why self-splicing introns does not require spliceosome? Because spliceosome is required, is taken, is needed by those who do not have the self splicing ability. So suppose if a strand, a DNA or something, it does not have a self splicing ability, then it will take the help of a spliceosome complex. But when introns which are self splicing, which have the ability to splice by their own, then they do not need the help of the spliceosome complex. Okay. So the answer of this question, this particular, is false. Then next question is, group 1 introns have an internal adenosine which targets the internal nucleophilic attack. So this question number F, yes, so Meghna is saying correct, that it self replicates, so it does not need it. Correct. Now this question, group 1 introns have an internal adenosine which targets the internal nucleophilic attack. I am putting this question on hold. First I'll tell you about it and then I will again ask this question. Okay. So we are already running out of time. So quickly we'll move to the week 2 content now. Okay. So week 2 was RNA as enzymes. So in lecture 1 we have studied about that. Basically in this chapter what we are studying, that how RNA can be used as enzymes. Okay. So what are the properties or what are the types of RNAs that can be used as enzyme? So which type of RNA can be used as an enzyme? Ribozyme. Okay. So have you in yesterday's class, have you also dealt with week 2 lectures? Yes or no? In yesterday's class, was week 2 also covered? Anybody? Please tell me. No. Okay. So that means in yesterday's class only week 1 was covered, right? So that means I have to give more time to week 2 then. Fine. So now we are seeing that how RNA can be used as an enzyme, or which RNA can be used as an enzyme. So RNA which can be used as an enzyme, that RNA is known as ribozymes. Okay. So in this lecture we have covered they have covered what is the structure of ribozyme, then what the comparison between ribozyme and protein enzyme. That means comparison between RNA, which works as an enzyme, and protein, which works as an enzyme. Then about in detail about ribozymes and protein enzymes and types of ribosomes. Okay, so up till now we all know that like whenever we talk about proteins, okay, so whenever we talk about proteins, we talk about whenever we talk about enzymes, we talk about proteins that works as enzymes, right? There is also one theory which says that all enzymes are proteins, but not all proteins are enzymes. Have you heard about this statement? All enzymes are proteins, but not all proteins are enzymes. There is enzymology. Yes, you must have learned. But now you see, so in biology, there are so many exceptions and there are so many things that you cannot give a statement like this. Now we are saying that RNA can also work as an enzyme. That means all enzymes cannot be proteins. Some enzymes are also RNA, for example, ribozyme. So next time when your teacher says that all the enzymes are proteins, tell them then what about RNA, ma'am? What about ribozymes then? Anyways, so, in the RNA world hypothesis, we can say that this RNA world hypothesis was the first to proposed as stages in evolution. Okay, so in this system, a variety of RNA enzymes could catalyze all the reactions needed to synthesize the molecules required for life from simpler molecules available in the environment, which is very important. That means in when the life was evolving, then there were not much protein enzymes available. Okay, so at that time the enzymes, that is the RNA enzymes were only the enzymes that catalyze the reactions which were needed to synthesize molecules. Okay, so that means this is one important thing, that initially when the world was originating, when simple molecules were forming, when simple molecules were combining to form complex molecule, then RNA was only the thing that was acting as an enzyme. Okay, so here it is saying that RNA is the only currently used macromolecule that is both a carrier of genetic information and an enzyme. It is very important. DNA is a carrier of genetic information, but DNA cannot be used as an enzyme. Protein can be used as an enzyme, but it is not a carrier of genetic information. But RNA, RNA can be used as a genetic information carrier also, plus it can be used as an enzyme also. This is a very good take home message that you should always remember, that DNA cannot act as an enzyme, protein cannot act as a genetic information carrier, but RNA, it can act as both. Fine. Then we come to the first type of catalytic RNAs that were discovered were ribozymes. Okay, so these ribozymes are the enzymes that are made of RNA instead of proteins. Very important point. So basically, what are ribosomes? Okay, then this was the first discovery, and second discovery was of ribosomes. And what are these ribosomes? These are large ribonucleoprotein complexes that translate RNA into protein. So why we are saying that ribosomes are also a type of RNA? Because ribosome requires the use of rRNA, that is ribosomal RNA, for the synthesis of protein, and also the enzyme which is required, which is taken use by the ribosome, that is also an RNA. Okay, that is why we are saying first enzyme, RNA enzyme is ribozyme, second is ribosome. What are ribosomes? Ribosomes are ribonucleoproteins. Then now there are very much available artificially constructed RNA molecules that can catalyze any kind of reaction, and then RNA sequences capable of simple forms of replications have been discovered. So basically these are all the evidences that shows that RNA can work as an enzyme. Now we come to the next part, that so basically the RNA world that existed. So these are the remnants of the RNA world. Now this slide is important, students. This slide is very much important. Okay, so this the like whatever points that we are dealing in these points in this slide. So a lot of questions will be asked for your assignments also. I think a lot of questions are there, and in general also. So what are the remnants of RNA worlds? You have self-splicing introns, you have RNase-P. So what is RNase-P? RNase-P is a ribozyme that cleaves tRNA precursors. Okay, you should always remember that RNase-P is a ribozyme which is specifically for cleaving the tRNA precursors. Okay, so these are the remnants of RNA world. That means at prebiotic era, RNA world existed, and now in today's world, these are the remnants which are still present in our environment, which are still present in today's world. Self-splicing introns are present in today's world. RNase P, which are the ribozymes that cleaves tRNA precursors, self-cleaving RNase, peptidal transferase. So basically the peptidal transferase enzyme, which is present in ribosome, which is used during translation, that is also an RNA. And then ribonucleotides or RNA, obviously, that is present. So now we come to what is ribozyme. Okay, so you see ribozyme. So basically ribozyme is a RNA that works as an enzyme. So when we see the structure of our ribozyme, so basically it has a primary structure. So primary structure essentially tells the sequence of the nucleotides, ATGC. So primary structure tells about the sequence. Secondary structure means that how this sequence has folded in itself. Okay, if this is the sequence, suppose, then how this sequence has folded. It has folded like this, like this, like this in two-dimensional space. Okay, then in three-dimensional space, the folding, then it becomes a tertiary structure. So when we talk about the primary structure, we're talking about the uh, the basis. That means the AUGC basis. Then this primary structure helps in the catalytic activity also. So basically ribozyme has the catalytic activity, and one very important feature of ribozyme is that it can help in the cleavage of the bond also and in the peptide synthesis also. These are the two important things where ribozymes are very specific, that it can cleave the phosphodiester bond also and it can help in the peptide bond synthesis also. Then we come to the secondary structure. So the secondary structure is basically the structure in the two-dimensional space which tells that how the primary structure has folded. This primary structure folded like this, like this, like this, or whatever. So you can see here, this is stem 1, stem 2, and stem 3. This is the cleavage site where the cleavage occurs. Okay, then there is a uridine containing turn. U-turn is there, uridine containing turn, and then stem 2 and stem 3, between them there is a augmenting helix. This is your augmenting helix that joins your stem 2 and stem 3. Is it clear? We are talking about the secondary structure of a ribozyme. So in a secondary structure of a ribozyme, it shows that how the primary structure has folded in the two-dimensional space. So you can see that there is a augmenting helix which joins stem 2 and stem 3. Okay, then tertiary structure. So tertiary structure, here it comes. So you can see that like I was talking about, that the divalent ions like Mg2 plus. So basically ions, they form coordinate bonds with your nucleotides. Okay, they form coordinate bonds and therefore they help in stabilization. It is very important to understand. Then you can see that this is the Mg2 plus site. The blue ones, these are the Mg2 plus site. Is it visible? In zoom also you can see these are the Mg2 plus site. So Mg2 plus is present in ribozymes. Is it fine to everybody? Are you following? Yes or no? Just type yes or no if you are following or not. Are you able to follow what I'm trying to tell you? Yes or no? Okay, fine. Okay, if you have any problem or if you do not understand, just tell me. Before solving the questions, I want to go through the literature part so that you can revise everything, because I know that in your hectic schedule of classes, it is very difficult to sit and properly read all the lectures. And that is why these online classes are conducted, so that you can get in just 2 hours, you can get a gist of all the chapters. And plus, we can solve some questions also. Fine. So then you can see, after this, there we will deal that there are certain naturally occurring ribozymes also. You can see these are the naturally occurring ribozymes. So tRNA is also a kind of ribozyme which helps in peptide bond formation, which is helpful in protein synthesis. Ribonuclease pre self splicing introns, group 1 and group 3. So basically these all are nothing but ribozymes. So tRNA, RNA, rRNA, tRNA, these are also ribozymes. Now we'll see the comparison of ribozyme and a protein enzyme. That means comparison of an RNA enzyme and a protein enzyme. So basically protein enzymes, why it happened that ultimately, just like in evolution, the genetic material changed from RNA to DNA. In a very similar way, the predominance of enzyme became protein enzymes rather than RNA enzymes. Okay, so RNA also have enzymatic activity and proteins also have enzymatic activity. But what happened that over the period of time during evolution, proteins become the predominant molecules that can be used as enzymes. Why? Because there is much more diversity when we take proteins as enzyme. Because RNA, we just have 4 unique nucleotides, A, T, G, and C. Whereas in proteins, we have 20 different amino acids. That means the diversity that protein can give as an enzyme is much more than the diversity that ribozymes can give. Second thing is that ribozymes have a negative charge. Okay, they have a high density of negative charge. So what happens that if a particular substrate is a negatively charged substrate, then it cannot catalyze because it will repel. Whereas there is no such thing with protein enzyme. For the charge of a protein depends on the type of protein it is, whether it is polar, non-polar, etc. Then so these are basically two very important points that leads to that why protein enzymes evolved as the predominant enzyme species. Okay, then these are the mechanisms. So you can see these are just you can just read, these are just the basics. Yeah, now this is very important slide, students. I am writing it here as important. Anyways, you are going to read about this in the next online class. But why I'm saying so, because in your assignment also, I don't know about this year's assignment, but this, for your exam point of view, so many questions are going to come from this types of ribozymes, especially in this thing chapter. So what are the different types of ribozymes? Metalloribozymes. So under metalloribozymes comes ribonuclease P and self-splicing introns. Then small ribozymes. Under small ribozymes, it comes hammerhead, hairpin, and hepatitis delta virus ribozymes. Then first so now we are dealing with types of ribozymes. Types of ribozymes. So first is metalloribozymes. Under metalloribozymes, which comes the ribonuclease P, RNase P. So we have already studied that RNase P, what does it do? It catalyzes the site-specific hydrolysis of precursor tRNA. That means ribonuclease P is required for the for showing catalytic activity in tRNA molecules. Always remember ribonuclease P, tRNA, ribonuclease P, tRNA. And it requires the activity of divalent cations such as magnesium and manganese. And this is about metalloenzymes. So we're talking about ribozymes. So the ribozymes can work as metalloenzymes, and in metalloenzymes, the first one is ribonuclease P. Okay, then you can see second comes so ribonuclease P, what we were talking about. So these ribonuclease P, what is the function of ribonuclease P? the hydrolysis of tRNA molecules. So it has two domain structure. In ribonuclease P has two domain structure. One is used for the recognition of the substrate and another is used as a ribosome active site. Okay. So first we'll deal some questions and then we'll do the second chapter onwards. And so we'll try to cover as much as we can. So I don't know whether we'll be able to cover the whole of the second week lectures or not. But if like at whatever point we'll stop today, I'll continue from there only in our next session. So in our next session, I will discuss about week 3 questions and some of the questions or some of the topics that we could not cover of week 2. Okay, both I will cover. So don't worry that if I'm not teaching today, then that portion is left out. We'll definitely cover it in the other class. Anyways, first we'll do some questions. Yeah. Okay. So tell me, a ribozyme is a ribonucleoprotein, ribonucleic acid, nucleoprotein, peptide nucleic acid complex. What is a ribozyme?
Ribonucleic acid.
Ribonucleic acid or a ribonucleoprotein?
Ribonucleoprotein.
Ribonucleoprotein.
Yes, correct. So a ribozyme is a ribonucleoprotein. Okay. Then prebiotic synthesis of sugar is via HCN, borate, Formose reaction, nitrogen base.
Farmer's reaction.
Okay. Correct answer. Is it correct answer? Prebiotic synthesis? Okay. Then Chargaff's rule says A is equals to G, C is equals to T, A is equals to
G is
C,
equal
G.
to C.
What is the correct answer?
G equal to C.
G is equals to C. That is the correct answer. Yes. A cannot be equal to C. A will be equal to T and G would be equal to C. Okay. Then next questions. We'll do some questions more. Yeah. No, this topic we have. Can you give the answer if you have you studied? Have you gone through week 2 lectures? Have you gone to anybody? You have gone through week 2 lectures? No.
No.
Okay. So this question we'll leave because this is in the subsequent class. Okay, we'll do this first. State true or false. RNA cannot store genetic information. True or false?
False.
False. Yes.
False, ma'am.
Yes, correct. Because RNA, it can store genetic information. Second question is, prebiotic world is a soup of RNA, amino acids and lipids. True or false?
Yes, ma'am.
True.
True, ma'am.
Okay. So are there only RNA, amino acids and lipids or there were some other um, molecules also? So this statement is actually not entirely correct. So we cannot say it completely true or completely false because prebiotic world, it does not only have RNA, amino acids and lipids, it has many other types of molecules also. But for here, we'll just consider it as true but insufficient, incomplete statement we can say. Then next question is, RNA is able to act as better enzymes than proteins. True or false? We have just discussed this. Can RNA act as better enzymes than proteins? True or
No.
false? False. Correct. Why RNA cannot act better enzymes than protein? Tell me why RNA cannot act better enzymes than proteins. Because proteins gives us more variability. Correct. There are 20 amino acids. 20 amino acid means so many different variety of enzymes can be produced. Whereas in RNA, you just have 4 nucleotides, A, U, G, C. So there cannot be much variability in RNA. And protein is a little bit more stable also than RNA. Anyways, then Any planet with hydrothermal vents can give rise to life forms. True or false?
False.
False. Why it is false? Because for the properly formation of a life, you just need you require a lot of more things. You just do not require hydrothermal vents that can give you heat. You required a lot of more things for the proper origin of life. Okay, then RNA molecule that catalyzes reversible cleavage and joining is, do you know the answer of this question? Molecule that can catalyze reversible cleavage and joining, yes or no? Okay, I am putting this question on hold right now because I think you have not studied about in studied the second this thing. Okay, let's see if you can answer some questions. I think you can answer these questions. RNA has a backbone made up of amide bond, ionic bond, sugar phosphate bond, or
Sugar
hydrogen
phosphate
bond.
bond.
Sugar phosphate bag.
Yes, answer is C, sugar phosphate bond. Now this point we have studied just now. Which is a metalloribozyme? RNase P, hammerhead, HTV,
RNA-P.
HIV. RNase P, correct. And what is the function of RNase P? Can anybody tell me? What is the function of RNase P? What is the function
Cleavage
of?
of tRNA
Yes, exactly.
precursors.
Exactly. So RNase P is required for the cleavage of tRNase precursor. So RNase P is a kind of a metalloribozyme. Okay. Now this again, first we'll study and then only I think you will be able to answer it. So we might discuss it in the next class or we'll see. Okay. A pre-tRNA molecule is matured by RNase A, RNase P, RNase D or base deletion. We have studied it. Pre-tRNA is matured by, tell me, what is the answer of this question?
RNase P.
Yes. RNase P. So see, this is same question is there. They have just changed the language. Here they are saying that okay. I was telling you that what is the function of RNase P? So RNase P basically cleaves the precursor tRNA, and by doing so, what it is doing, it is actually causing the maturation of precursor tRNA. So always remember that RNase P, it cleaves the precursor tRNA, and therefore pre-tRNA gets matured. Is it clear? So, pre-tRNA molecule is matured by RNase P action. Now, cofactors in ribozymes are important for why? Why cofactors are important? Because these cofactors, they stabilizes the RNA secondary structure. Okay? Always remember that the secondary structure of RNA are stabilized with the help of cofactors. Okay? The peptide bond formation during protein synthesis in ribosome is by, tell me, peptide bond formation during protein synthesis in ribozyme is by,
tRNA.
tRNA.
okay, more questions, sorry, more answers. Hmm, correct answer, correct answer. Then we come to next questions. Hmm. This question we have studied, the stem 2 and stem 3 of a ribozyme is joined by, we have just studied this. Tell me, what is the answer? Stem 2 and
Augmented
stem
helix.
augmenting helix. Correct answer. You can see here. We have just studied about this. Ah, sorry. See, stem 2 and stem 3, and this region is augmenting helix. Augmenting helix joins stem 2 and stem 3. Okay. Then next question is, sorry. Yes. Metal ions responsible for the stability of ribozyme structure is calcium ion,
Magnesium.
magnesium ion, phosphorus, sulfur. What is the answer?
Magnesium ion.
Magnesium
Magnesium
ion.
ion.
Yes. So magnesium ion is responsible for the stability of ribosomes. Okay. So we have studied lecture 7. Now we'll come to lecture 8. Quickly we'll see what is there in lecture 8. So we have studied about ribonuclease P. So ribonuclease P is basically required for the hydrolysis of tRNA, and in that it matures the tRNA. Now this ribonuclease P has two domain structures. That means the domain has two sides. One is required for the substrate recognition and another is ribozyme active site. Then second type of metalloribozymes are self-splicing introns. Here you can see here when we were studying about ribozymes, the different types of ribozymes we were studying. So you can see that ribozymes can be metalloribozymes or small ribozymes. Now metalloribozymes can be ribonuclease P, which are required for the maturation of tRNA, and the other one is the self-splicing introns. Now what are self-splicing introns? Basically self-splicing introns are large intronic regions which are able to splice by themselves. What are introns and what are exons? You know what are introns and what are exons?
Yes, exons are
Non-coding
coding
sequences of
region, introns are non-coding region.
Yes. So basically, introns are the non-coding regions and exons are the coding regions. So basically, so self-splicing introns are the large intronic regions which are able to splice by themselves. And then you can see introns. What are introns? These are the non-coding RNAs. And before translation, you can see when transcription happens, you can see these are the exons and these are the intronic regions. Now, intronic regions are not required for in the mRNA. So these regions are spliced out and only exons they join together. Okay, you can see that only exons are joining together. This is a matured mRNA. Okay. Now we want to compare a self-splicing and a splicing process. Okay. So basically, not all the introns are self-splicing introns. So self-splicing introns can splice themselves without the need of a spliceosome complex. This was a question also that was there when we just solved this question. That introns which are self-splicing, they do not need a spliceosome because spliceosome is basically a complex which is required for splicing. Self-splicing introns does not need them. Okay. So this is very important point which you should understand. And self-splicing introns mediate only one round of RNA processing. Now, there are two types of self-splicing introns, group 1 introns and group 2 introns. You can see that group 1 introns uses a guanosine. So basically it takes use of an external guanosine. Whereas group 2 introns, they have internal presence of A. So this was the question that you were not able to answer earlier. Uh, where was that question? Huh. So can you answer this question now? Excuse me. Question number F. Group 1 introns have an internal adenosine which targets the internal nucleophilic attack. It is true or false? Can you tell me now? Can you tell me? False. Why? Because group 1 introns have what does group 1 introns have? They have G nucleotide. So group 1 introns have G nucleotide, which is an external guanidine nucleotide, whereas group 2 introns have internal
Yeah.
adenosine nucleotide. This they take from outside. This G nucleotide comes from outside, and this A nucleotide is present in the group 2 introns. So the question is, group 1 introns have an internal adenosine? No. Group 1 introns takes the external guanidine, and both guanidine and adenosine are required for nucleophilic attack. Okay, then this is all about the self splicing introns and the mechanisms. Then we come to small ribozymes. So we were talking about the ribozymes types. So ribozymes can be of two types: metalloribozymes, which includes ribonuclease P in self splicing introns, and small ribozymes, which are hammerhead, hairpin, and hepatitis delta virus. Okay, so these small ribozymes are basically satellite. What are satellite small ribozymes means? They are smaller ribozymes which kind of helps as an helper virus in helping the multiplication or infection. So one example is satellite virus. So what are satellite? These are small RNA virus, and their multiplication depends on the mechanism of host cell or co-infection of the host cell with the helper virus. So I will stop here so that I will explain these two uh, lectures in the upcoming class, because these are a little not difficult, but I think they require a little bit more assistance. So you should understand that these ribozymes, they can exist as two parts. Small ribozymes are basically those ribozymes which are smaller. Not only smaller, they have accessory function. That means they do not function completely on their own, rather than they help in the assistance of some bigger molecule. So they help in the proper functioning of some bigger molecule, and that is why they can also be called as satellite. Why they are also satellite molecules? Satellite means small helper things. So you can see that these satellite are the small RNA virus or small RNA molecule whose multiplication depends on the co-infection with another virus. Okay, so basically this is what I'm trying to say. So this was a question you can see here. um, okay, so we'll deal it in the next class maybe, because it will be a lot of for you to retain. So we'll stop here. And so we have covered the lecture 1, whole lecture 1. We have revised it, and we have revised half of the week 2 classes. So in week 2 there are 3 classes, and we have revised half of the week 2 classes. And questions, I think most of the questions we have already done. So in the next class we'll revise half of week 2 and then completely of week 3. Okay, so in this way we'll move. I'll, because my aim is not just to make you solve questions, but also to make you understand the concepts and whatever is there in your theory, so that you have a basic idea about it. So do you have any questions? You can ask me, or just tell me that is this way is fine for you, or should I only teach questions, only solve questions, no theory, or you want theory plus questions both? You tell what you want.
Theory plus questions, ma'am.
Okay, so other, you want theory plus questions both or only questions? Please tell. Both. Okay, so I will do it like this only, theory and questions. I will both, I will simultaneously I'll do. Okay, so one topic we'll study and then we'll solve two questions from that topic. Then another topic we'll study and we'll solve two questions from that topic. So with that, it is easy to understand topic more efficiently, madam. Yes, I can understand, because we all have such busy hectic schedule. So you might not have that much of time to give entirely on the syllabus. That's why it's helpful for you to just revise once the topics that are there. So fine, so with this I will stop today's session. Hope you liked it, and if you have any queries related to anything, I am typing my mail ID. So if you have any questions, any queries, you can mail me on my mail ID. Fine. So thank you. We'll stop the session now. Okay, bye bye.